All right, welcome back everyone for another Build a Cell seminar. Thank you so much, Masayuki Imai, for agreeing to give a seminar presentation today. I will, as is Build a Cell tradition, let you introduce yourself and then take it away.
Okay. May I start my talk?
Yes.
Okay, so thank you very much for Carries. So I am Masayuki Imai from Tohoku University. Well, first of all, I would like to express my special thanks to Kate giving me these opportunities. Okay. My background is soft matter physics, and current my research topics is uh, mainly the minimal cell or proto cell and the morphogenesis of multicellular organisms. Okay, so today I would like to talk about a synthetic minimal cell from soft matter physics point of view. Okay, this figure shows the proliferation of bacteria, where bacteria takes in nutrients from the outside and converted the nutrient to the molecules that constitute themselves. So such using such molecules, the bacteria shows growth and division as shown here. That is a proliferation. This proliferation is quite unique and never observed in non-living matter systems. So, great mystery is that how did the living system emerge from matter? That is the question. Okay. This figure shows a long history from the simple molecules to the multicellular organisms. So initially, the simple molecules are converted to the a little bit complicated molecules, such as amphiphilus. Amphiphilus start to self-organize to form the self-assembly, such as vesicles. Simultaneously, some genetic molecules appear and start to produce proteins. Proteins are couple of some chemical species and form the metabolic networks. In the metabolic networks, uh, uh, eventually form the membrane molecules, and the membrane molecules are incorporated into the vesicles. And then vesicles shows growth and division. In this stage, we call the protocell stage. Eventually, the protocell evolved to the first living system for unilamellar organisms. And further, the such unilamellar organism evolved towards a multicellular organism. That is a contemporary living systems. So convenience, today I would like to, for this side is physical world, and this side is biological world. Okay, in the physical world is governed by non-equilibrium thermodynamics, where the entropy change is divided to the two parts, that is entropy change in the reversible process and the entropy change in irreversible process. The time evolution of the such non-equilibrium system is determined by the minimization of the entropy production in irreversible process. That is a physical world. On the contrary, the biological world is evaluated by the fitness. That is a reproductive success. And the living system evolved towards the peak of the fitness landscape with time, as shown here. The problem is that I would like to integrate these two concepts in one world. My For this purpose, first, I would like to focus on the protocell. But unfortunately, it is quite difficult to realize a protocell in our experimental setup. So instead, we would like to focus on the minimal cell. Minimal cell is a model cell systems with a minimal essence of living systems. Okay, so what is the essence of the living system? This is a schematic drawing based on the Shoes papers. So where the living system takes in nutrient glucose from the outside, and the glucose is converted to the energy currency molecules, ATP, such as ATP, in the glycolysis pathways. The ATP encourages the replication of DNA, and the DNA produces the proteins. Using the ATP and this molecular machine, the proteins, so this system synthesizes lipid using a byproduct of the glycolysis, that is a glycerol 3 phosphate and the pyruvic acid. Synthesized lipid is incorporated in the vesicle membrane, and the vesicle membrane shows growth and division. So this is a simple expression or essence of the living system based on the Shoes paper. And then the living system is composed of mainly the three important units. One is the energy molecule production unit, and the second one is a genetic polymer processing unit, and the third one is a lipid synthesis unit. Okay, based on this essence of living system, we designed the synthetic minimal cells. Okay, this synthetic minimal cell mainly composed of the AOT molecules. This is amphiphilus composed of a sulfate head group and the two alkyl chains, okay, and form the vesicles in a water solution. Then we supply glucose from the outside. The glucose is converted to the hydrogen peroxide with the enzyme GOD. So hydrogen peroxide plays a role of the energy currency molecule in this synthetic minimal cells. So we call this part is energy production part unit. The hydrogen peroxide react with a monomer of the information polymer, in this case aniline, and produce the aniline radical cation. So in this case, aniline radical cation has four types structures depending on the position of the radical cations. So type A is a radical cations located at nitrogen atoms, and the type B is located at the para position, and the type C and the type D are located at also position, as shown here. Here, the AOT molecules play an important role. Without AOT molecules, without AOT vesicles, these four species randomly react and produce a polyaniline with random sequence, something like that. But in the presence of the AOT molecules, so between the AOT and the aniline radical cation forms a hydrogen bonding, something like that. Due to this kind of hydrogen bonding, so only the type A and the type B monomer units react and form this kind of linearized polymers. That is a polyaniline emeraldine salt, or we call it PANI-ES. So having this kind of specific sequence. This kind of polymerization we call the template polymerization due to the restricted by the hydrogen bonding. So AOT, PAN-ES on the membrane interact not only the vesicle membrane AOT, but also the AOT in a solution. So AOT molecules trapped by the PAN-ES increases the hydrophobicity due to the blocking this sulfonate group, and then this AOT molecules in the external solution incorporated into the vesicle membrane. Then vesicle membrane show growth. That is a simple picture of our synthetic minimal cells. And due to this kind of selectivity, we call this polymer is information polymer. And this section is an information polymer processing unit. And this part is a membrane growth polymer, where the AOT molecule are incorporated in the vesicle membrane. So let's see to examine our design works or not. So in this case, we prepared AOT vesicle in the solution and then supply the uh, glucose, that's the nutrient of this synthetic minimal cell. And also we supply aniline and enzymes from the second mic micropipette. And from both pipette, we supply the glucose and the aniline. And what happened is observed by the optical microscope. So here I show the movies. Okay, so this is a target AOT vesicle, and we supply nutrients and monomer of the information polymers. So initial 40 seconds, it does not happen. Nothing happen. But around the 40 seconds, the vesicle start to fluctuate and shows growth and deform to the porolate cell, as shown here. And this is a snapshot. You can clearly show the growth of the vesicle. And this is a time evolution of the surface area with the time. So red one is obtained from this kind of experiment. So clearly the vesicle area increased exponentially. Now these data is a control experiment without polymerization. So you can see the significant promotion of the vesicle growth by the information polymer PAN-ES. Okay, here, then we examined similar effect for the other type of amphibians. For example, we prepared phospholipid vesicles, DOPC, and this is a neutral phospholipid, and a negative phospholipid DOPA, and the AOT is a sulfonate group, as shown previously, and the SDBS, another type of amphibians with a sulfonate head groups. And we prepared four types of the GUVs and inject, supply the DOPC, DOPA, AOT, and SDBS, and check the growth is occurred or not. The black circle shows that no vesicle growth was observed, and the red circle indicates it shows a significant growth. So from this table, we can understand vesicle growth is specific to the sulfonate head group amphiphilus. And so then the AOT vesicle catalyzes the synthesis of the PAN-ES, and the PAN-ES promotes AOT vesicle growth. This selectivity is the essence of this synthetic minimal cells. The one big advantage of this synthetic minimal cell is the chemical scheme of this system is well described. For example, energy production pathway from glucose to hydrogen peroxide is described in this way. And the synthesis of information polymer, PAN-ES, is described in this way. And the uptake on membrane AOT molecule towards the vesicle membrane is described by this kinetic. Then all of the chemical reaction scheme is described precisely. Based on such chemical scheme, we can construct simultaneous differential equations to describe the time evolution of each component. For example, hydrogen peroxide is described by this equation, where the V is a reaction rate. And the reaction rate for the V1 can be described using reaction rate constant, something like that. Similarly, aniline radical cation creation is described by this equation, and the polymer chain and the polymer segment and the AOT vesicle area is described by this equation. And the all reaction rate is described by here. Okay, so by solving such an equation, we can predict the growth or synthesis of the information polymers. This is a comparison between the theoretical prediction and the experimental result. So this figure shows the membrane growth part. Okay, so normalized surface area increases in this way, as shown before, and the theoretical prediction is that this red profiles, red curves. In this case, the important point is all of the rate constants can be obtained from the literatures or independent experiments. That means that no adjustable parameter. All parameters are already fixed. So without such parameter fitting, we obtain a very good agreement between the theoretical prediction and the experimental result. Similarly, so funny ES synthesis is also described by this data. So the experimental data will describe as a previous kinetic model. So surprisingly, so not only such outcomes, we can express the consumption of the nutrients. For example, in the case of the consumption of the glucose is well described by the previous kinetic models, and the consumption of aniline monomer is also described by the kinetic models. That means our designed kinetic scheme is well realized in our synthetic minimal cells. Okay, so far, so I showed our designed chemical reaction pathway for the synthetic minimal cell well works. But unfortunately, vesicles shows only the growth, not the reproduction. So we have to evolve towards the reproduction. So reproduction means that the spherical vesicle shows a growth and then deform to the limiting shape. Limiting shape, the two spherical vesicles are connected by the narrow neck. And then this narrow neck is destabilized and shows a division. So these two daughter vesicles should be recovered the original size. That means original area and the original volume. So this is a point for the sustainable reproduction. Next, we would like to discuss this part. So vesicle deformation is described by the so-called membrane elasticity model. The free energy is described by this equation, where the kappa is bending modulus, and the C1 and the C2 are principal curvatures, and the C0 is a spontaneous curvatures. So spontaneous curvature is 0 means that membrane prefers a flat geometry, and non-zero means that membrane prefers a bend geometries. Okay? And kappa G is a Gaussian curvature modulus. Okay, so the vesicle deforms to minimize these elastic energies. Okay, so this is a morphology diagram proposed by Seipert. And here the various vesicle morphology is plotted against reduced volume and reduced spontaneous curvatures. Reduced volume is a measure of the volume to area ratio and defined by this equation, where V is a vesicle volume and A is a vesicle area. R is a vesicle area based radius of the vesicles. Okay? So reduced volume is 1 means the vesicle has a spherical shape. And decreasing the 1, the vesicle has an excess area. Such excess area, using such excess area, vesicle deform to the porolate, limiting shape, porolate, pier, oblate, and stomatocyte. And the vertical, here the vertical axis reduced spontaneous curvature. This is a spontaneous curvature normalized by this vesicle radius. Okay, so when we prepare the vesicles with a one component amphiphilis with a cylinder shape is a has a head group and the bulky tail has a almost same size. So in this case, the membrane has a no spontaneous curvatures. Okay, in this case, vesicle the spontaneous curvature is 0, then vesicle deform to the porolate as shown before. But in order to attain the deformation towards a limiting shape, vesicle should have the spontaneous curvature. A simple way to produce induce a spontaneous curvature is to mix a two type of the amphibians. Here we mix the inverse cone shape lipid. Okay, inverse cone means a small head group and bulky tails. Due to the such geometrical preference, the inverse cone shape lipid prefers asymmetrical in the bilayers. Such asymmetry causes the spontaneous curvature as shown here. So due to this spontaneous curvature, basically deform to the limiting shape. This is our strategy to attain the such basically deformation to the limiting shape. Next, we consider the vesicle division. In order to attain the vesicle division, we should satisfy two requirements. Okay? This is the energy landscape. Stage one is a limiting shape, and stage two is a two vesicle state. In order to vesicle divisions, first, such energy barrier between the limiting shape state and the two vesicle state is overcome by the thermal fluctuation, thermal energies. And second, the free energy of the two vesicle state should be lower than the limiting state, limiting shape state. Okay. So here we express the free energy model for binary vesicle composed of the cylinder shape lipid and the inverse cone shape lipid. The total energy can be described in this way. This is the previous one. And here, as shown before, due to the coupling between the lipid shape and the spontaneous curvature, this is a non-zero. It causes a deformation to the limiting shape. Here, the important point is the coupling between the limiting shape and the Gaussian curvatures. In the case of the cylinder lipid, kappa G, this is the Gaussian curvature modulus, has a larger value than the inverse cone lipid, kappa G prime. So here the limiting shape vesicle has a spherical part and a neck part. So spherical part has a positive Gaussian curvature, and the neck has a negative Gaussian curvature. So the cylinder lipid prefer to stay in the neck region, and the inverse cone shape lipid prefer to stay this spherical segment. So due to the such segregation, they stabilize the neck, and the vesicle division is attained. This is some kind of theoretical considerations. Here, we check the such a vesicle division using a inverse cone shape lipid by the molecular coarse grade molecular dynamics simulation. Here, we prepare the cylinder shape lipid with the same with the size of the head group is almost same as the tail, single tail. And also that we prepare the inverse cone lipid with a small head and a bulky tail. Now we prepare the two vesicles. One is a single component vesicle composed of the cylinder lipid, and another one is a binary vesicles composed of the cylinder and the inverse cone shape lipid. And check the stability of the limiting shape or pear shape. So, okay, so first single component vesicle transform to the limiting shape. It has a very narrow neck as shown here, but this narrow neck is quite stable. On the other hand, in the case of the binary lipid, it's developed to the limiting shape gradually. Yeah, as shown here. So you can see that this neck is quite stable, but in the case of the binary vesicles composed of the cylinder and the inverse cone shape lipid, this neck is quite unstable, and soon such a neck is broke and produce a two vesicle state. But still, the neck is stable. That agree with the theoretical prediction. Okay, so based on these arguments, we try to we try to realize the reproduction of the synthetic minimal cells. In this case, in addition to the previous AOT molecules having a cylinder shape, we add the second component, cholesterol, with a very small head groups. We mix the two types of the lipid, and then performed a similar experiment. That is, we surprise AOT molecules and glucose and aniline molecule. That is a monomer of the information polymer. And check what happens. And, okay, so you can see the mother vesicle produces daughter vesicles, and then daughter vesicle produce granddaughter vesicles, and granddaughter vesicle produce great granddaughter vesicles, as shown here. Okay, it seems works. But when we plot the surface area of the vesicle and the volume of the vesicle against the time, this is the time evolution of the surface area and the volume. You can see the surface area, the blue point, increase in this way, but total volume is almost constant. This shows that this system is not a sustainable reproduction system. Then we have to increase the volume of the vesicles. Then a simple way to attain such vesicle volume growth, that is the inflation of the vesicle, is using osmotic pressure difference. For example, first we use sucrose. That is osmolyte impermeable to the AOT membrane. So we encapsulated the sucrose inside the vesicles, and then this with a concentration of the 100 millimolar is transferred to the sucrose solution with a concentration of 90 millimolar. Due to the osmotic pressure difference, the vesicle shows inflation. But in this case, the inflation is quite limited due to the very fast inflow of the water to the vesicle membrane. So, due to the such inflow of the water, the concentration of the sucrose inside the vesicle is immediately diluted. In order to avoid such a quick dilution, we use a asymmetric osmotic pressure technique. That is, we use two osmolytes having a different permeabilities. That is, in this case, we encapsulated sucrose that is impermeable to the membrane, and also we use a fructose. This is a permeable to the AOT membrane. So we encapsulated sucrose inside the vesicles with 100 millimolar. On the outside, we use a fructose with 100 millimolar. In this case, not only the water, but also the fructose is also permeate into the vesicle membrane. Then in this case, so volume inflation continues very long time compared to the single osmolyte case. And it decreases it shows a somewhat sustainable, long period vesicle inflation. Okay. Integrating such techniques, finally, we examine the recursive vesicle reproduction of the our synthetic minimal cells. Here, you can see our AOT vesicles and the supplies or component by the micro injection. Okay. In this case, again, the mother vesicle produce daughter vesicles. In this case, with elapse of time, the mother and the daughter vesicles grows not only the area, but also the volumes. Okay, so this is a snapshot of the such recursive vesicle reproduction. So initial vesicle has a unit area and a unit volumes. So when it's divided to the two vesicle state, the area of the mother vesicle is 0.91 and the volume of the mother vesicle is 0.86. On the other hand, the volume area of the daughter vesicle is 0.77 and the volume of the daughter vesicle is 0.68. But after 100 seconds, so area of the Okay, sorry. Area of the mother vesicle developed to the 1.2 and the volume is 1.4 around 4. And the daughter vesicle area is 1 point around 1 and the volume is also 1. It recovers original volume and the original area. Okay. So then here, this is the statics. Okay. For example, first we prepare around 50 mother vesicle and examined this kind of this kind of experiments. Okay, so about 45 of the mother vesicles produce the daughter vesicles, and about 22 of the daughter vesicles produce the granddaughter vesicles, and 14 of the granddaughter vesicles produce great granddaughter vesicles. But it seems about sustainable vesicle reproduction attained. Of course, the failure of this sustainable recursive vesicle reproduction is due to the we cannot supply the appropriate concentration of the ingredient because we use a manually micro injection technique. If we can do that more ideally, this ratio might be increases, but I think a little bit difficult for the manual operation. Okay, so here we compare the heart of the living system and the heart of the synthetic minimal systems. Okay, heart of the living system is a central dogma where the nucleic acid sequence in the DNA is translated to the amino acid sequence in the proteins. And this amino acid sequence determines the catalytic nature of the protein to produce the lipid molecules. And since and the molecular basis of this central dogma is a hydrogen bonding between the adenine and the thymine and the guanine and the cytosine. This base pair is causes a molecular recognition by the hydrogen bonding. Okay, okay. And due to this central dogma, synthesized lipid molecules are incorporated into the membrane, and the cell membrane shows the growth and the division. On the other hand, in our synthetic minimal cell is a base, molecular base is a hydrogen bonding between the AOT membrane molecules and the aniline radical cations. And due to this hydrogen bonding, so AOT membrane produces uh, polyaniline emeraldine slot, PANI-ES, having this specific sequence. Also, due to this specific sequence, so AOT PANI-ES interact with AOT molecule in the external solution and controls the chemical potential of the AOT molecule in the external solution. Then this AOT molecule incorporated into the membrane, and the membrane shows the growth and the division as shown before. That is the heart of our synthetic minimal cells. Okay, so now I would like to introduce our ongoing project in our synthetic minimal cells. First one is exploring the new species to examine the evolution by the selections. Now I show our synthetic minimal cell that is a PANI-ES and AOT system. So in order to attain such a competition and selection by competition, we need another species. Here we are now trying to examine the new species, polypyrrole and the SDBS system. SDBS is an amphibious, is a similar sulfonate head group and a bulky tails, and produce some kind of this kind of vesicles. And the polymer is a polypyrroles. That also shows a template polymerization on the vesicle membrane, on the AOT vesicles or SDBS. So we examine the this candidate for the new species for the synthetic minimal cells. The experimental setup is shown here. Here we prepare the mother vesicles composed of AOT or SDBS. And the supplies monomer of the polymer, that is aniline for PANI-ES and the pyrrole for the polypyrrole. And also we supplied ingredient AOT or SDBS. Then we have the 8 species. That is a 2 times 2 times 2 times. And measures growth of the vesicle using a light dynamical light scattering technique. Okay? In this case, the vesicle is a SUV with a size of the radius of around 50 nanometers. So obtained dynamical structure factor is fitted by this kind of a single exponent function. And from this relaxation rate, we can obtain the hydrodynamic radius using a Stokes-Einstein relationships. Okay, so here I show the vesicle growth rate for the various species. Okay. Horizontal axis is a supplied AOT concentration or ingredient concentration. And the vertical axis is a membrane growth rate normalized by the initial vesicle size. Okay? This figure is a mother vesicle is AOT, and the polymer is a PANI-ES, and the ingredient is AOT. And the red data is with polymers, and the blue data is without polymers. You can see the clear promotion of the vesicle growth in the presence of the PANI-ES. This is also a similar experiment, but the mother vesicle is SDBS, and the polymer is polypyrrole, and the ingredient is AOT. In this case, we also observe the significant increase of the vesicle growth rate. But in the case of the AOT vesicles, polypyrrole polymers and the AOT ingredient, so such promotion effect is mild. And in the case of the SDBS, PAN-ES, and the AOT, such a promotion effect is quite limited. Okay? So this shows that the vesicle growth rate depend on the type of the species. Based on these experiments, we construct growth landscape or fitness landscape of our synthetic minimal cells. Okay, so growth rate is expressed by the size of the spheres. So big sphere, big sphere means a large growth rate. Okay? And the red color means that we observe the significant vesicle growth, and the blue color means that promotion is negative, suppressed. This axis means the type of the mother vesicle, AOT or SDBS, and this axis shows the type of the polymers. And this one is PANI-ES, and this is a polypyrrole. And this axis indicates type of the ingredient that is the AOT and the SDBS. So you can see that the growth rate strongly depends on the type of the species. And the highest peak is species 1, species 6, and species 7. And the lowest one is species 4 and species 8. Then we are now planning to experiment to examine the selection by the competition by mixing of these type of the synthetic minimal cells. Another project is to examine the non-equilibrium thermodynamics of synthetic minimal cells. As shown before, the chemical scheme of the synthetic minimal cell is well described with this kind of the scheme, such as a nutrient glucose is converted to the energy currency molecules, hydrogen peroxide. Hydrogen peroxide is transported to the membrane surface and react with aniline monomers and produce the aniline radical cations. Produce aniline radical cation react each other and produce polymer, polyaniline emerald salt, PAN-ES. PAN-ES encourages the incorporation of the AOT molecule in an external solution to the vesicle membrane. Okay? This time evolution of each component can be described in this differential equation. And the important parameter is the vesicle growth rate, lambda, is expressed by this relationship and expressed by this equation. K for one, the rate constant, reaction rate constant for this process, and P2 is a concentration of the PAN-ES polymers. Okay, as shown in the introductions, we have two requirements. One is a biological requirement that requires a maximize reproduction success, fitness. But here this fitness is approximated and maximizing basic growth rate, RAM. On the other hand, the physical requirement of the requirement is a minimizing entropy production. So using based on these two requirements, the total requirement is that so maximizing basic growth rate while minimize entropy production. Then we will check this concept is works or not. Fortunately, we well described the chemical scheme of our synthetic minimal cell. We can calculate entropy production of the synthetic minimal cell, as shown here. This is the entropy production. This part is the entropy production for the formation of the aniline radical cations. This part is the entropy production by the formation of the polyaniline emeraldine salt. And this part is the entropy production, the transportation hydrogen peroxide from the external solution on the membrane. Okay, and this entropy production reaches a minimum at the non-equilibrium stationary state. That is expressed by these equations. Using these constraints, we can express the growth rate as a function of the nutrient concentration, glucose concentration, and as a reaction rate constant. Then we can plot the growth rate landscape or fitness landscape against the nutrient concentration and the reaction rate constant. This is one example. The vertical axis is a basic growth rate, and this axis is a glucose concentration, and this axis shows a reaction rate constant K41. You can see the higher concentration of the nutrient and the higher reaction rate constant K41, the greater the basic growth rate. Okay? It seems that this result is very reasonable for the synthetic minimal cell, but a little bit strange because if the concentration of the nutrient is very low, if the system has a higher reaction rate constant, such a cell consumes the nutrients rapidly and starve. That means such minimal cell cannot survive in such a very low nutrient concentration. So we propose a new evaluation function, K function, is described in this way. So this K function is a balance between the reaction rate constant and the entropy production. Okay? And the C is a balance constant. For example, if the system has enough glucose nutrients, C is quite small, then the basic growth rate is a priority. On the other hand, very low nutrient concentration, C is a very high, then in this case, entropy production is priorities by suppressing the basic growth rate. This is some plot with such examples. Okay, so if the glucose concentration is high, the balance constant is 0.1. Then the K function is plotted against the reaction rate constant, in this case, K31. It shows a monotonically increase with a K31, which is consistent with the previous result. Okay? But when the starved state, that is a very low glucose concentration with a high balance constant, so K function has a maximum at a very low reaction rate constant. That means, so in this case, so this system minimize energy dissipation by suppressing the vesicle growth. Okay, so this result is consistent with the strategy of the living system. Unfortunately, we are not derived the really physical basis of this K function, but it seems a good evaluation function. Anyway, so finally, I summarize I'd like to summarize my talk. So now we are developing a synthetic minimal cell with a similar chemical scheme or the living system. Okay, so this synthetic minimal cell cannot synthesize the membrane molecules from the byproduct of the energy products in it. Instead, the supplied membrane molecules are incorporated into the vesicle membrane by the using a information polymers. But this system shows sustainable growth and division. That is the present stage. And the remaining challenge of the synthetic minimal cell is shown here. One is the autonomous synthetic minimal cells. That means we would like to synthesize AOT molecule from the byproduct of the energy production unit. That is a gluconic acid. We would like to synthesize AOT molecules from the gluconic acid. That is a big challenge to approach the synthetic minimal cells. Second one is evolvable synthetic minimal cell. This means a fluctuation of the template polymerization may produce a new synthetic minimal species, and the result of the selection and competition, it starts to evolve. That is the ideal cases. And the third one is I would like to reveal the physical background of the living system, such as K function, using a information thermodynamics. Okay, so this is a collaboration of our project, and mainly this work was done by the Minoru Chris, this guy, and also supported by the Peter Valde. He is an expert in the template polymerization, and Yuka Sakuma, she is a specialist for the vesicle division. And Taro Suzuki is now charging on the new synthetic minimal species. And Professor Kawakatsu and Takagi are now charging on the theoretical part. And then Professor Urakami doing a numeric molecular dynamics simulation. Oh, thank you very much your attention.
Thank you. Wow, very exciting and very impressive work, I'm sure. We have some questions in the chat already, and then I invite anybody else to raise your hand, unmute yourself, or post your questions in the chat as we go. Um, so
Can you
I
hear
can get the
me?
ball rolling and read this question for us. Mohammed asked, why is the growth of the AOT vesicle making the vesicle longer, i.e., not radially expanding? What can cause that asymmetry? With a follow-up
Can
comment,
you hear
he
me?
says, yes. Can you hear the questions? Should I reread it?
Alice, can you hear me?
Yeah. Can you hear me? Oh, no. Can everybody else hear me? Okay.
Sorry, I cannot hear. Alice, can you hear me?
Yeah. Yes.
Sorry, I cannot hear your voice.
Hmm. Hmm. Hmm.
What, what happens? Yeah, I can read chat. So, okay, so what to say? I can read the chat. But Mohammed, why is the growth of the AOT vehicle making the vehicle longer? Is I not really expanding? What can cause that asymmetry? What is G- can you hear me? Okay, so this question is why the vehicle developed was a hollow shape, not a spherical shape, I think. It's okay? And in this case, the volume of the vehicle is constant, but the area of the vehicle increases. So in such a case, the reduced volume is decreases, and depending on the spontaneous curvature, it's preferred to the hollow shape. Is it enough
Yeah, I'm sorry you can't hear
for
us.
the
I'm sorry. Let's see. Does anybody else have questions you can post in the chat?
is okay?
This might be where we end up wrapping up.
Ah, okay, I can hear it.
Oh, okay. We're back
I'm
online. All right. It didn't last long. I like that. Um, let's see. Sorry, everyone, for those brief technical difficulties. I'm not sure what happened there. Does anybody else have any follow-up questions? I know Mohammed responded to his own saying that your kinetic models match the data. Do you include this in your model somehow? And this is responding to the vesicle shape. Does the model suggest elongated vesicles?
okay, so your, your kind of model still match the data. Do you include this in E- Yeah, in this case, we include the shape of the vesicles. Here, we
Okay.
extract the surface area and the vesicle volume by approximating the vesicle morphology using a surface evolver. That is some
Oh,
uh, vesicle models. Then we can extract the vesicle area and the vesicle volume. And the
okay.
data is then using such a data, we plot against the time.
Okay. That asymmetry is really interesting, though. I was curious about that myself. Let's see. Does anybody have any last questions for Masayuki before we wrap it up? We'll hover here for a second, but thank you so much for giving. That was a wonderful talk. And
I'm
my
sorry for
goodness,
some technical problem.
it didn't last long. We got through it. It was wonderful, though. I have a lot of notes to review and a lot of things that I'm excited to learn more about. So it was wonderful. And thank you everyone else for attending. This will be posted on our YouTube channel in the next couple days, but we'll review it first. All
Yes.
right. Thank you. Have a great day.
Thank you very much.